And as with everything, there's optimum conditions. So this is an example of the optimum condition for an enzyme. And the blue line is showing that we haven't yet achieved the optimum condition for the normal enzyme. But for the temperature sensitive enzyme, we can see that our optimum temperature is at its peak. So our enzymatic activity is highest at the peak. So again, enzymes have optimum temperatures. And an optimum is the temperature that they work best at. Here's an example of a graphical presentation of an optimum pH of our enzymes. So our enzyme pepsin, which is represented in the blue, has an optimum because of the peak, pH of close to 1, whereas trypsin or glycogen phosphorylase have optimum pHs at higher values, at approximately 7 for glycogen phosphorylase and at approximately 8 for trypsin. So before we can get into some more detailed metabolic reactions, we need to look at metabolic pathways. And metabolic pathways come in two forms. In essence, a metabolic pathway is taking your reactants and eventually forming products, but you have intermediates along the way. So if we look at the top, we have a linear metabolic pathway. We start with our reactant or reactants. They are turned into product by an enzyme. Notice the enzyme is written above the arrow because it's not part of the reaction and it's reused after the reaction. The product from our first reaction becomes known as an intermediate when it becomes the reactants, in this case, of course, substrates because we're talking about an enzymatic reaction, for the next series of reactions. So reactant 1, energy of activation lowered by enzyme 1, has a product formation that then becomes the reactant or substrate for enzyme 2, so it's called an intermediate. And then that product becomes the reactant or substrate for enzyme 3, so it's an intermediate. And then we get a final product form. Notice this is a straight shot, that's a linear pathway. We can't have a cyclic pathway, and that's what we're seeing down here at the bottom. And notice the cyclic pathway is in a circle. And in a cyclic pathway, your reactant enters an enzymatic reaction, forms a product that becomes the reactant or substrate for the next enzymatic reaction, so it's called an intermediate, which is catalyzed by an enzyme, enzyme 2 in this example, forming our product that becomes a reactant, so that's becomes an intermediate, which is catalyzed by enzyme 3. Now when enzyme 3 catalyzes that intermediate, some of the product returns to become the initial reactant, and other product formed actually becomes the product and is used in some other cellular process. Notice the major difference between the top and the bottom of this slide is the top is a linear pathway, whereas the bottom is a cyclic pathway. We're regenerating our starting compound in the cyclic pathway, but not in the linear pathway. Now there's also such a thing as called feedback inhibition, which is a form of regulation of metabolic pathways. So imagine we need a product, but we don't need to just continually make the product because then we'd be wasting our resources and our energy. So here we see our reactant being catalyzed by enzyme 1 into a product that becomes the reactant or substrate for enzyme 2. That means that it was an intermediate. The product of enzyme 2 becomes the reactant or substrate for enzyme 3. And then we have a final product formed. So this is a linear metabolic pathway. That final product can then go back and turn off the first enzyme. And when it turns off that first enzyme, it shuts down the pathway because with enzyme 1 not working, the first reactant cannot become the first intermediate. And that intermediate can't become the second intermediate, and that intermediate can't become our final product. So we're regulating the pathway. When the product concentration gets low, our enzyme will no longer be inhibited, and the whole pathway will turn back on. So this is a way that the cell regulates how much of a specific product it is producing so that it doesn't overproduce that product. Now, of course, we could have chemical reactions occur and they would release an enormous amount of energy, so much energy that our cell, which is performing a specific type of metabolic process, would not be able to use all that energy and it would just be wasted as heat. So cells use those pathways to help control product formation and control energy production and use. So this slide is in essence just comparing to the left, breaking glucose down outside of a cell, and to the right, breaking glucose down inside of a cell. So when we burn glucose outside of the cell, we immediately get our carbon dioxide and water, and we liberate energy, but it's all released in one big shot as a bright light and heat. Well, we don't want that to happen in a cell because it would be too wasteful. So a cell has multiple enzymes, multiple enzymatic pathways, where it will step by step take the energy of the glucose, break it down, forming carbon dioxide and water. But it will do it such that the energy is going to be released slowly and in packets so that it can be used by the cell. And we do have waste in the form of heat when the cell breaks down glucose, but it is much, much less than if we just burnt glucose in a crucible like we see in this picture. Now, for all of these chemical reactions to be able to occur inside of a cell, we need to move substances in and out of the cell. And one way to do that is by the process of diffusion. So in this slide, we see some food coloring added to a glass of water. And that food coloring added to the glass of water is going to diffuse outward from where it was originally added. So we can see at the perimeters, it's very clear because we have not yet had diffusion of the original substance that we added in the center. So you can see here in the center, the substance is very, very concentrated, and out here, the substance is much, much less concentrated. Now over time, the substance in the center will diffuse all the way out so that the color of the container is all that uniformly pink color. So diffusion is in essence movement of a substance down its concentration gradient. So in essence, concentration is how much of something like a solute is dissolved in a given amount of a solvent. So originally, what was the concentration of the pink indicator? How much of the pink indicator was added to the container of water? And a concentration gradient is established by having different concentrations between adjacent regions. And that's best illustrated in this next picture. So over to the left, we have a U-tube, which is basically just a glass tube, and it has a selectively permeable membrane at the bottom. And it's filled with equal amounts of solution on both sides of the U-tube, that's the blue. But on the U-tube side to the left, we see fewer red dots, that's the solute. And on the right, we see more red dots. So the concentration to the right is higher. We have more red dots or solute and a set amount of solvent than on the left. On the left, we have fewer red dots or solute and the same amount of solvent. So our concentration is lower on the left than it is on the right. So we have a concentration difference. So that means we have a concentration gradient established between the left side of the U-tube and the right side of the U-tube. If we let time pass and the only thing that could move was the water, which is blue, our water would move from where we have more solute, which was on the right, to where we have less solute in order to dilute the solution. So a quick review. In this image to the left, we have a concentration difference or a concentration gradient set up by our amount of solutes in our solvent. Since we have more solute in the U-tube or the half of the U-tube to the right, that's a higher concentration of our solute or red dots than we do in the U-tube to the left. That means we have a concentration gradient. Now the reason why in the picture to the right we don't see the red dots moving is because this selectively permeable membrane does not enable them to move. So they can't. But water can move. So since water can move, it is going to move to help equalize that concentration difference. So if you can't equal out the red dots by the red dots moving, you can equal them out by doubling or adding more water. That's the end of the presentation for Chapter 5. I hope that it has been of help.